Welcome to The Chain, the podcast exploring the lives, careers, research, and discoveries of protein engineers, scientists, and biotech professionals. We look at the impact their work is having on the field and where the industry is headed. Tune in to stay up to date on the newest advancements and to hear the stories that are impacting the world of biologics.
Hello, everyone. My name is Tarek Bayoud, and today I'm having a conversation with Professor Peter Tessier, Department of Pharmaceutical Sciences and Departments of Chemical and Biomedical Engineering, University of Michigan at Ann Arbor. Dr. Tessier and his team are developing novel approaches to expedite the developability of biologics. We will discuss his science, of course, but first, Peter, tell us about your scientific journey. I would like to know more about your background and how you got to where you are today, with special emphasis that when did you decide to become a scientist? Were there any specific events, conversations, mentors that guided through this journey of yours?
Great question, and thanks for this opportunity to chat. I think it started in high school for me. I was really inspired by chemistry. I loved the idea of trying to understand the molecular world. I did my undergraduate in chemical engineering, and I never really thought about going to graduate school until one day a mentor of mine just said, "You should go to graduate school." And I thought, maybe I should go to graduate school. And that sort of was, the turning point for me. And when I was a graduate student, I got to be around somebody. His name is Orlin Veliv. He's not known in the biological field, but if you look him up, he is a colloidal scientist and he comes from Bulgaria, and one of the great minds I've ever worked with. And it's super creative, and creativity is an interesting thing here when we think about scientific journey, because for me today, I feel like I'm still trying to live up to the creativity of this mentor of mine. He was just an amazing person, and that really inspired me then to want to go on into academics. And sort of the rest is history, as they say. That turning point for me was graduate school, being around just an incredibly creative scientist.
Were you always at University of Michigan, and how did you end up at university?
Yeah, exactly. Good question. So, I went to, so I did my PhD in chemical engineering, and it was, I was doing biophysical sort of experimental work, but I really wanted to learn more about biology. And so I went and did a postdoc in a lab at this Whitehead Institute of Biomedical Research at MIT. And I was a fish out of water, as they say. So I knew very little about what I wanted to, I didn't know a lot of molecular biology or cell biology. Just an example how much I didn't know, I actually applied for an NIH fellowship, and I got the highest score my lab had ever got, but it was because when you read the reviews, basically the reviewer said, "This guy doesn't know anything, and he has a huge training potential," so they scored me very highly. But for me, that was a really important point to jump out into a new area and learn a lot of molecular biology and cell biology that really today has been very influential. So I went on and then started my independent academic career. It was about 16 years ago now at, it's called RPI, Rensselaer Polytechnic Institute. It's an engineering and science school in upstate New York. And then about 5 years ago, I moved from there here to today. Now I'm at the University of Michigan. So that's kind of what brought me here. That's right.
Very nice. Yeah, that's a nice story. I've been learning new things as you go from your graduate work to your postdoctoral training, and I think that's what it's all about. So just to move on from what you just said, that, science is a team effort, and having a motivated team is really critical. Having people around you who are motivated and inspire you and things like that. So the question I have for you is that, how do you motivate your team now that you have graduate students and postdocs? So what is your
Yeah.
How do you work?
Well, that's a good question, and I think one that a lot of us that have teams of people that we're trying to lead, that we ask ourselves a lot. So for me, the first thing it starts with is I'm personally very excited about science. It's the thing that I just love doing the most, and so I first want to set the tone for just the infectious curiosity and excitement about science. So that's the thing I love the most. That's the thing I spend my precious time most on. And so I want to create that environment around me that just this is what I love and this is what I do, and join me in that. I think the second thing is that we try to foster in our lab. There's no hierarchy. So if you're an undergraduate student with a good idea, you know that you get as much. Your ideas are valued just like anybody else. So I think the idea generation and the enabling people to be creative and to come up with their own ideas is really important. So when I tell a PhD student when they come in the lab, the most important thing that I hope to accomplish here is by the time you leave, you're an independent thinker and you can really drive your own research. And so I try to motivate through infectious interest in science, and also just a sincere interest in helping people develop to become independent scientists.
So, you described some really key elements of motivating people, and then the other thing is that it is really also important to have a certain core traditions or culture within the lab, so to speak. So, do you have any core traditions or things that you do in the lab that keep people together and thinking together?
Sure. I mean, it's humbling talking to you here because you're the one that should be teaching me about this. But I think some of the core traditions that we have are, first of all, try to have ownership. We want people to have ownership over projects. So I don't want to be the one deciding everything we do. I really want ideas to come from within. So one way we try to do that is we try to do science together. So I don't meet with people just one-on-one on a regular basis. We try to meet in small groups because we want to facilitate thinking together, reviewing data together. And usually a lot of the best ideas don't come from me. And, I try to have this core culture that we do it together, and that we think about together, we plan together, we gather ideas together. So, I think that's probably one of the most core aspects of what we do. And then the other thing is that, it's a self-motivated enterprise. So core tradition is that I don't go checking on people if they're in the lab or not, but that it's simply self-motivated because we want to instill ownership, self-motivation, and really have people be able to determine their own fate and really have ownership over things. So, I think those are two. I think a third one would just be integrity, that we never want to publish something that we don't believe in. We want to be, extra careful about as we do science, be very self-critical, to make sure that we get it right. So those are some of the things that come to mind.
That's really nice. Yeah, I think those are the critical values. I think in training people, in mentoring people, those are really, really important aspects. So, just to continue on this topic, so as you know, in research, one has to deal with both successes and failures.
Yeah.
And it is really important to keep that in mind, and especially when you're working with younger people in your group. So, how do you deal with number one, first, successes, and then failures, which is really important?
Yeah.
If you could give an example or a couple of examples of each, that would be really great.
Sure. Yeah, so this is it's such an interesting and important question. So I think maybe the first thing I would comment on is that, as you have to embrace failure, and being willing to fail to do science well. I mean, that's, generally, we and you and others work on problems that are hard, and they're not, if it was easy, somebody else would have done it. And that means that often it takes time, and you have to fail. So I think that that's one thing that, first of all, we try to embrace in our lab, is that it's okay to fail. You try to often have multiple plans in case you fail, but just being okay with that. In different cultures, as I mentor students, some students that come from certain cultures are less comfortable with that. And so, I think that it's something that I have to continue to work with. Some people get it easier than others. But it's such a core thing in science that really, if you want to do innovative science, you've got to be ready to fail a lot. And interestingly, I think really success is the ability to, keep going. So I think that I admire the students in the lab, and a lot of them are good at this, that just keep going even despite, say, repeated failure. I think, maybe one example that comes to mind that's been both a success and a failure is, we're very excited right now about delivery of biologics to the brain using bispecific antibody technology. And without worrying about all the details, what I can tell you is that I have a PhD student who is finishing after about 5 years. In his first few years, he started all this work in our lab, was extremely humbling, to the point where if he didn't have sort of this really sort of optimistic personality, I'm not sure we would have made it. And we started off targeting one thing at the blood-brain barrier, and we just weren't getting anywhere. And then we switched into something that is involved in amino acid transport. And that has been incredibly fruitful, and probably the one of the most exciting things I've ever been personally involved in. And so, I think that process, one of the things that you have to ask yourself as a scientist when you're failing is, first of all, do you believe in the idea? Okay, or the general, even without worrying about all the details, do you think you're going in the right direction? In making judgments about whether to keep going in that direction or not. And of course, everybody has to deal with this, but I think that one of the things that I found is that if I'm working on something that I don't really believe in, or I'm not sure about the importance, or whether it's really that feasible, sometimes you go through the motions trying to fix a problem, but you're not really committed enough long-term. Where, for example, in this brain delivery project, we knew we were committed for the long term. We believed in the idea, and it kept us going during the times where it was incredibly frustrating and humbling. I mean, on a day-by-day basis, if you evaluate your career in one day, you would feel really bad about yourself. But if you could, keep that horizon, that, we don't know how to do this, or this experiment is not working, but we still know or strongly believe we're going in the right direction, that then sort of sustains you. And that's been a big thing to sort of go through failure to finally come out on the other side.
Yeah, those are really, really interesting stories that you just tell. I can fully appreciate those. So just continuing, because this is such a fascinating topic, subject in research. So was there an idea that you really felt or you really believed in, and ultimately it did not pan out, that it still lingers in your head?
Yeah. These are painful memories, and some of them I flushed out of my brain. I think when I think about that question, one of the things that comes up is that, as a scientist, you often ask yourself, are you working on the most important problems that your skills and abilities and the potential impact that it could have, are you doing the right thing? And I think some of the things that hurt the most when I look back at what I would call sort of failures was spending time working on certain things that in hindsight were not very important, and a lot of them were very hard to do. When I finished my postdoc, I had worked on these yeast prion proteins, and we tried some very difficult experiments for like several years. And in hindsight, the exact experiments we were doing were really not important enough to justify all the effort, but they sort of made sense at the time, even though if I could go back, I would sort of change the focus. So I think those are the parts that really haunt me in a way. And I think all of us, are we working on the most important problems that we can work on that have the greatest potential impact for science, for fundamental science, for therapeutic applications, and so on? And still today, that's really something that I struggle with on a regular basis, is that, are we doing the right things? Are we working on the right topics with the greatest potential impact? And that's a hard question to ask, and a question I think all of us have to keep asking ourselves.
So yeah, no, that's really great. Thanks for sharing that. So just to continue this conversation, so as we all know that drug discovery and development, especially small molecules and biologics, is a long process. And identifying, especially in biologics, developable antibodies during the early discovery phases is really critical. And in the past few years, you and your team have been in the forefront of trying to develop methods. And there are some really interesting methods that you have come up with in terms of developability. So can you talk about this work? How did you arrive at that, decided that is the critical question, as you were alluding to earlier, and how did that work progress with this AC-sins and CS-sins and polyspecificity and all those, because this is such a critical, important issue for drug development.
Yeah, great questions. Well, it's really been our pleasure to be part of this important field. A field that you've worked in a lot longer than I've worked in, but we've been fortunate to be part of this exciting antibody therapeutic explosion that's happened over the last few decades. And as you say, as lots of antibodies are being generated, people want to know more and more, earlier and earlier, is this molecule going to have all of the drug-like properties that we want, so we don't waste any additional effort or have any problems in making it and producing it and formulating it and keeping it stable and getting the pharmacokinetics that you want? So that's really motivated people to ask, what is it that's really important, right? What are those important properties that we should be looking at about an antibody that would tell us as much as possible about how it's going to behave, especially later in development when we're kind of too far in to start changing anything, right? You want to know that as early as possible when you still have that opportunity to make changes to the molecule. And there's a lot of things that are important. We focused on a few of what we think are particularly important, but we're not trying to claim these are the only things that are important. But what's been fascinating to us, there was sort of a landmark study published in 2017 by Adimab, where they did, at the time at least, the most comprehensive kind of biophysical characterization of approved antibody drugs and also things that were in clinical development or had failed in clinical development. And they looked at a range of biophysical properties to ask, were any of them superior for approved drugs compared to those that, say, had failed or were still in clinical development? For those of us that have been around for a while, we know about Lipinski's rule of 5. Think about this was sort of starting to try to think about, is there some antibody equivalent to that? Like what would matter? So for those of us that know Lipinski's rule of 5, there's an octanol water partition coefficient. That's, a measure of hydrophobicity. You could say, is there some equivalent to something like that in antibody world that might tell you, is a molecule too hydrophobic, or is a molecule, for example, does it have, self-association or instability, folding stability, or whatever it might be? And interestingly, when they did that analysis, there are things that came out to matter, or at least to be superior for approved drugs, were not obvious. At least they weren't obvious to us. Like, we would have guessed, if you would have told us that they were doing this study, we would have guessed, well, approved drugs are probably less aggregation prone. Approved drugs might be less hydrophobic. Approved drugs might be more stably folded. Like, all of these things that are kind of relatively obvious compared to, say, things that had failed. Like, maybe the things that had failed were just, they just had some of these problems. But interestingly, the only things that turned out to be statistically significant were measures of what you could kind of loosely call non-specific interactions, or colloidal interactions. Sometimes people would think about them. So think of things like self-association. So these very weak non-affinity interactions mediated by the variable regions, or just non-specific binding, meaning the antibody binds to things it shouldn't bind to, non-cognate antigens. And that was kind of an eye-opener, because out of the 12 things they looked at, 5 of them were significant, and they were all these sort of non-affinity, weak colloidal interactions. And that really opened our eyes. Now, truth be told, we were already working in this field, so we were quite happy to see that what we were working on happened to be important. So that was kind of fortunate for us. But it really started to catalyze a deeper appreciation. Wait a minute, there's something really interesting here that even we were really not fully appreciating. And so, that has then led us to even invest greater in this area to try to do a couple things that, couple goals that we would have. One would be is we want to have experimental methods, and we and others, not just us, that are really good at measuring these weak interactions really early in the development, and I would say discovery and protein engineering stages of drug development. And the biggest thing that you need assays that work at low concentrations. We call these ultra dilute concentrations for biophysical measurements. So these are typically on the order of 10 micrograms per mil, for example. So for us, we think of that as basically, if you can measure the affinity of a protein, of an antibody, then we want to be able to measure these biophysical properties, particularly self-association and non-specific binding. So that's what we've been doing there, is trying to develop those kinds of assays.
So, Peter, that is really exciting what you're doing. But one of the things that is really critical in early discovery, as you just mentioned, that when you're working with a very large number of molecules early on, and you need to identify your lead candidates, let's put it that way. So what are you and your team doing towards handling large numbers of these molecules?
Yeah, good question. Yeah, so absolutely. I suppose this is a task you've done many times, these discovery campaigns. And so when you have this, you have lots of molecules, like you said, early. So therefore, there's 2 things we're trying to do. So there's the experimental part, and then there's a computational part. But just on the experimental part, therefore, what you just brought up, the assays need to be easy to run in parallel, right? You need to be able to measure multiple things at the same time or very quickly, really to be amenable to some form of high throughput screening. Now, high throughput is thrown around loosely a lot. So one thing that might be helpful is just to say, what we want to be able to do is measure hundreds to thousands of different antibody molecules in an assay that's relatively straightforward to run. You just want to be able to run it in well plates, for example. So just 96 to 384 well plates in a very standard system. So, for example, the nanoparticle-based assays we've developed can just be run in well plates, just using a standard spectrophotometer. So they're that's easy to apply to relatively large numbers of molecules. Our non-specific binding assay is a flow cytometry assay. Again, those are very commonly available with plate samplers, so they're easy to run in a plate mode as well. We think this is really important to be able to do exactly what you said. The other part's the computational part, and ultimately we'd like to displace all the experiments, and so that's a separate issue.
Yeah, so that we will come back to the computational part. But so just continuing on this topic, the assays that you're developing. So, these days within the field of biologics, there are so many different formats that people are developing. And the assays that you are applying and you are developing now, you have tested those for just the antibodies so far. So what is your opinion, or how do you see these assays being applied to other formats like biospecifics, ADCs, or nanobodies? Are you working on that, and do you think you would need modifications in the current assays?
Yeah. Great question, and something actually we're really excited about. And so big picture, we focus primarily on IgGs. And you're bringing up the important point that there's a lot more than IgGs today. In fact, it feels like you and your company and your industry are coming up with more formats than we can keep track of. But I'm joking. But nevertheless, there is a lot of bispecific, multi-specific, nanobodies, as you say, and VHH, other formats. So we are starting to work on that area. We're really looking, though, for people to work with. So that's something that is a big interest to us right now. The assays that we've developed, I believe, will be readily adaptable to lots of different formats. Now, there is going to be some, adapting that needs to be done. So some formats are easier for us and some are harder. So we start with, if it has an FC domain, it's pretty easy to imagine how to readily adapt it, because we're generally binding the FC region in our assays to sort of capture the antibody. And if it doesn't have an FC, we have ways of doing that as well. But I think this is a particularly exciting area. One of the reasons we're so excited about it is that it's probably even more important, because a lot of these non-standard formats have more problems. For us, that's more interesting. For people like you, it's more annoying. I'm joking a little bit, but like for academics, this gets us excited, because this is the kind of thing that you guys have got too good making a lot of good molecules today. So now these are like a new challenge that present lots of interesting challenges that really, therefore, this developability analysis is even more important. I would say the problem is it's also less developed, right? There's just not the understanding right now for the developability of these non-standard formats as we have for IgG. So there's really a gap there in knowledge that needs to be addressed.
So just to continue on that, Peter, so different organizations, different groups, they develop all kinds of assays to select their lead candidates. All right. And there's a lot of information out there in the literature. So with your work and your experience in characterizing these antibodies, what do you think, or in your opinion, what are the most critical steps and what are the nice to have steps? What that means is, because if we can somehow identify, based on your work and other work, the most critical aspects of moving the programs forward, that would really impact the timelines, which is really critical. So what's your opinion?
Interesting. Yeah, good. Those are good questions. So essentially, we're thinking about what's critical here? What are the properties that are most important, or the assays that are most important to run? So from my perspective, there's some kind of foundational things that you just have to have, that you have to know about a molecule. So I think that size exclusion, characterization of the percent monomer of the protein, it's sort of, we take it for granted, or it might seem trivial, but actually, especially for non-standard formats, looking at percent monomer, say initially, and then over time in different storage conditions. So, cold, room temperature, 37 degrees. This is a very helpful foundational piece of information that you want to have. The folding stability of a molecule, critical. There's some properties that the industry has got so good at performing that we don't see problems when we get to clinical stage molecules. So things like that, foundational. I think there's some sequence-based liabilities that are things that you can look at on the sequence level, but they should not be underappreciated how important they are. So looking for chemical degradation sites, glycosylation sites, and so on. And we still see on a relatively routine basis as we look at things that are in different stages of clinical development, particularly for molecules that have been developed some time ago, there's lots of different liabilities that are still around that people have still not, fully. So I think there's some of those they're just so foundational. If you have a chemical degradation site in the CDRs that's going to be, or a glycosylation site that's going to make analytical characterization, I think those are foundational. I think when you start to think about then what would be nice to have, I think that versus what's critical, I think one question would be is, as the field is advanced, if we think of something like formulation, do you want your molecule to fit into a platform formulation? Do you want to minimize the amount of time that you're going to be spending on formulation? I think to the extent that that is increasing desire, there's really a big emphasis on platform formulations, trying to make molecules fit very standard formulations. Then I would say that looking at things like self-association early, because there's also such a push to subcutaneous delivery that, if you have to have it at, this high concentration and it has to fit a platform formulation, I would say looking at self-association early fits in somewhere between absolutely got to have it and it would be nice to have, somewhere in there. And I think the more the field goes along, perhaps it'll become, critical. I'd say right now we're probably somewhere in between critical and nice to have. The other thing that is, that we think a lot about is pharmacokinetics. So do you want your molecule, do you want to reduce the risk of having abnormal pharmacokinetics due to, for example, non-specific binding, so non-target mediated effects, right? Not set the target mediated effects aside. Antibodies and other types of multi-specific antibodies can have abnormal pharmacokinetics, fast clearance, for example. So I think looking at non-specific binding is another one, again, which historically is not in the critical bucket. It's been in the nice to have, but if you have that problem, abnormal pharmacokinetics, it's a disaster, right? Because you can't, you're stuck. You can't change the molecule by the time you're measuring pharmacokinetics. So I think that, that's another one that's not historically been in the critical bucket, but is really commonly done today. And I would say, again, it's probably fitting in between, critical and nice to have.
So Peter, like some of the methods that are being applied, you mentioned a long list of assays that people use, but do you think that applying this polyspecificity assays and looking at self-associations at low concentrations and things like that, the assays that you are developing, that these assays would help us not do a whole bunch of things, but if we can bring these assays up front. And the reason I ask that is because, as you mentioned about other liabilities and sequences, sequence informations, now people are making advanced libraries. So they can incorporate some of the features in the libraries and then other features by applying the methods that you are developing to really shorten the timeline. Do you think that is possible in that context?
I think that's a very, very exciting developing area that I totally agree with you. The goal overall, like you say, shorten timelines, which is code for do less experiments, right? So, to shorten the timeline, we've got to need to do less experimentation, and we've got to make it so that, it's more predictable what's going to happen. So that that's really the critical part here. So I think, like you said, so for for the library design and, and some of the work that's being done there, that's definitely going to be very helpful to eliminate some of those liabilities so we never even see them, right? And then, but it's still very difficult to get it all right, and ultimately there's got to be some experimentation that's done, partly just to reduce risk overall. I mean, we know some, but we'll never know everything. So, there'll always have to be some experimentation. I mean, there are some people in this world that won't believe any computation, just saying. And so, you always have to have an experimental step to make sure that the molecules have the properties. But of course, trying to minimize the number of experiments that are needed, trying to do them early to avoid problems later, I agree. So there's a combination of the design and the computation. So that could be the library, like you said, one part of that could be the library design. Another part is just analysis of antibodies wherever they're coming out of, right? Because some, of course, are, library, in vitro libraries, but others are just, coming out of animals. And so having really computational methods that can identify those molecules that have the properties that you want to be part of the filter process, I think is also a critical piece.
So you brought about the computational design. Just a quick question that these days, this big data and machine learning is a big thing.
Mm-hmm.
and the assumption is that the more data we collect, the more we will learn. So, combining all that. But the question I have for you is that what are the critical problems that we can solve with this kind of computational information right now? Do you think that down probably 5 years, what we can solve now or what we can solve 5 years from now, and what kind of data do we need, right? So right now, there's a lot of emphasis just in collecting sequences, for example.
Right.
But the assays and the data that you are generating and others are generating, that has not been incorporated fully.
Right. Yeah. I mean, this is like $1 million question you're asking here. I mean, there's a lot of pieces to it. So I think one part of it is what could computation do to advance the field that we're really not able to do right now reliably. So, I think that there's part of it, which is if we think about, from an antibody perspective, what are some of the, say, the affinity, the targeting of the antibody, say, to specific epitopes on a molecule, when we think about sort of the biological, targeting and activity of the molecule, it is difficult to target some epitopes that we want to target on molecules using experimental methods. There's immunodominant epitopes that, when we stick an antigen into an animal, that a lot of the antibodies will go to certain sites, and some of those are not what we want. So I think that just on a wish list, if computation could increase the ability to target predetermined epitopes on antigens, that's a pretty exciting potential advance. So that's an area where, I think computation, it's not there fully yet. I mean, there are some examples that are spectacular today that are already being demonstrated, and you can imagine that it's just going to, further advance. So I think that type of design or redesign, people call it different things, that's something that the field needs. We need more predictable targeting of epitopes on target. Some antigens are incredibly difficult to make. I mean, you know this better than I do, that are very difficult to generate antibodies against, because in many ways, we often think of the antibody discovery problem as the antigen problem. Some antigens are just really poorly behaved, or they don't behave the way you want when you take them out of their biological context. So could computation advance the field by enabling us even to get binders? We don't need high affinity binders against every epitope. We need lead molecules that then target the region we want, that then could be, so I think that's one area. Okay. Now, another area that computation is impacting, when what we're talking about is improving, design and even filtering of antibodies that have all of these developability properties that we want. And I think that that's an area where there is a lot of potential. And one of the things that comes to mind about that area is the design for affinity or antigen binding. It's hard because everything matters, like down to the atomic precision of the prediction. It's a very hard problem. Now, there's progress being made, but in the developability world, if we think of things like self-association, non-specific binding, hydrophobicity, it's not that everything matters to the same atomic resolution. So I do think there's a lot of potential for computation. I'm not saying it's easy, but we're finding that, developing methods, we and others, and there's a lot of other people, for predicting viscosity or predicting, some of the properties, some of these developability properties. There is a lot of potential there, that one could do with computational methods. So I guess those are going to continue to kind of, be integrated together, that we're going to be designing things for the activity we want and the developability we want as we merge those together. And they probably are going to go at different pace. I didn't address your question about data. That's a really tough one.
But do you think that, there's a lot of data being generated, but do you think that data sharing amongst different organizations and groups, is that to your liking at this point?
It's just a huge, I mean, you know this, it's a huge problem, and I think at the heart of it, the problem is that there is the data is very valuable. Okay, so if you're an organization and you go and you generate, some of this really precious data. So what in our world, viscosity data is very precious. So if you go and generate viscosity data for a panel of antibodies, especially a large number, say 50 to 100 molecules, that is a significant resource investment just because of the sheer amount of protein that you need. And when you have that information, that's a competitive advantage today you can develop models using this data. You can start to improve your drug development process. So I think that, data sharing is hard. Again, there's lots of different kinds of data, like you said, everything from just human antibody repertoire sequences to, biophysical characterization of high concentration antibody formulations. So some of this data is easier to share because it's less unique. It's less precious. It's less expensive to generate, and so on. So that's the gap right now that we're finding, is that the really precious data for the developability part of the problem, especially that's difficult to generate, it's not being shared so that each organization is essentially doing it themselves. And so you have a lot of effort that is being duplicated right now.
Yeah. So Peter, thanks for sharing your thoughts on the science and all that. So I have a final question, let's say. So you are definitely, you have been very successful in science and motivation and how you train people and all that. The question I have for you, what are your hobbies or interests other than science? And
Yes.
number one, and out of this, if you were to do things differently, what would you do?
Yeah. Wow. Well, let me take the softball one first. So in terms of hobbies, so I am so I have a dog. I never had a pet until about just before the pandemic. We finally bought a pet, and I never thought I'd love pets, and now I want to touch every dog I see. So I got a dog, and now every dog, I'm like in a cult now where every dog is amazing, and I'm totally out of control. So I love my dog, and so my wife and I we walk like all the time. So we're like prolific walkers. So walking is definitely just in nature. We live in Michigan in kind of a rural area, so we do lots of walking, which is almost like hiking around where we are, and that's with our dogs. So that's one of my favorite hobbies to do. The other thing is I'm a crazy sports fan, so I'm always following. If I can only open one web page in this whole world, it would be ESPN.com. So I'm always checking. It doesn't matter what sport it is, I'll follow it. So I've been following World Cup soccer right now very closely, and so those are kind of some of my hobbies. I have a family and kids and everything, so I don't have tons of hobby time, but that's my extent of my hobbies. And yeah, that's an interesting point about what would you do differently in your career if you could kind of go back. I think one thing that I still am trying to do on a daily basis is that doing science is not easy. And one of the things that happens is because failure is frequent, it's kind of a nerve-wracking business to be in. And if I could do one thing differently, it would be worry less about kind of the future, worrying about if things don't work out, what am I going to do? I think I've spent too much time in my career worrying about, because it is nerve-wracking. It's not easy when you're going through times of sort of failure to know, is this going to be okay? Am I going to, and I think I wouldn't want to change my passion and my intensity for science, but I just wish I could have worried less. And I think each day now, because sometimes it takes away some of the joy. I think that it's such a joy to be involved in science, and sometimes worry and fear, which come from being on the edge and trying things that are hard, they can eat away at your joy. And every once in a while, I just can't believe I get to be on a podcast with you talking about science and antibodies. And, it's crazy. I mean, this is the stuff that we love and that we get to do as our job. It's quite amazing, right? So
Yes.
If I could worry less, that's the one thing I would do. And I need to try to keep doing better at that.
Well, that could be your next year's resolution, New Year's resolution.
There you go. You got it all lined up for me. Perfect.
Okay, Peter, thanks so much for your time and sharing with us all your lessons learned, experiences, and science, and above all, your acquired wisdom. So, yeah, and moving the field forward. So, we wish you all the best and success, and all the best in 2023.
Thank you so much. And the same to you. I really appreciate this.
Thank you very much.
If you would like to attend any of CHI's upcoming protein engineering conferences, use the discount code TheChainPodcast, all one word, to save 10% off your event registration.